Hi everybody! I'd like to invite you to a video clip in which I'm going to help you better understand the various events that occur at a typical synapsis between two consecutive neurons. So let's take a look at the sketch that I have on the screen here. What I have labeled as presynaptic is an axonal terminal of one of the many axonal terminals of a presynaptic neuron, a neuron that sits prior to a synapsis. This is then the cell body with dendrites and axon of a postsynaptic neuron. Ultimately, the axon of that postsynaptic neuron will split into many axonal terminals that in turn may yet again synapse with another neuron if we were inside of the central nervous system, or possibly these axonal terminals could be synapsing with some kind of an effector, such as cardiac muscle cells, skeletal muscle cells, smooth muscle cells. Let's come back to the presynaptic neuron. Inside of the presynaptic neuron, we have in the purple synaptic vesicles filled with their orange neurotransmitters. We have all along the dendrites and the cell body membrane, and you can assume all along the axon these blue squares which represent chemically gated ion channels. Chemically gated ion channels will open up and allow for the flow of ions, either positive or negative ions. It depends on the neurotransmitter and the receptor. When a neurotransmitter binds to them. That's why they're called chemically gated, because they respond to the binding of a chemical, a neurotransmitter in this case. And like I said, these chemically gated channels can occur everywhere, even along the axon. The reason for this is that it can happen that a synapsis occurs. Let's say I could add this in our drawing. We could even have a synapsis between a presynapse synaptic neuron here and this axon, or even here between an axonal terminal here and this axonal terminal of our current postsynaptic neuron. So we have to have chemical gated ion channels along the axon as well. But more often than not, we're going to see either a dendrosomatic, a dendrosomatic synapsis, somatic referring to the, I said that wrong. I meant to say we're either going to see an axodendritic synapsis, meaning a synapsis between an axonal terminal and a dendrite, or we're going to see a synapsis between an axonal terminal and cell body, in which case it's called an axosomatic synapsis. And this axonal terminal actually functions as both an axodendritic as well as an axosomatic synapsis. These green elliptical structures I drew along in the membrane of our axon represents our voltage-gated ion channels. Voltage-gated ion channels only open when there is a significant change in voltage right at the level of the axon hillock. And we can assume, it's not the best drawing I made, but let's assume that right here is our axon hillock. And that's where we would see our first series of voltage-gated ion channels. Typically, there aren't any, or not many, voltage-gated channels in any of the dendrites or along the cell membrane of the cell body, so that we will not see action potentials firing and propagating in the dendrites in the cell body. So the action potential is going to start at the level of the axon hillock, typically, and then it'll propagate, and we will learn what that means today, all along the cell membrane of our axon, all the way to the various axonal terminals. On the other hand, we have graded potentials occurring in response to the binding of neurotransmitters to the chemically gated channels. These neurotransmitters can eventually reach the axon hillock and possibly trigger the firing of an action potential. Let's take a look at all of this. So let's go back to, well, let's add one more thing, I'm sorry. Let's also remind ourselves that this presynaptic neuron has an action potential propagating down all the way towards the axonal terminal. This action potential will function as a stimulus to open up yet another type of gate, and let me use the color white, and let me just make this as just a cross line. These would be specifically calcium voltage gated channels, while the green ones are going to be both sodium and potassium voltage gated channels. So in the axolemma here of our axonal terminal, we have many calcium gated channels. I apologize, that's my little puppy playing with a toy. And so when the action potential arrives at the axonal terminal, we're going to see that these calcium voltage gated channels open such that calcium, which is high in concentration on the outside of cells compared to the inside, calcium will literally flow into this synaptic vesicle. When calcium does that, various biochemical reactions take place, and ultimately what will happen is that the synaptic vesicles go through the process of exocytosis, and this leads to the release of the neurotransmitters. These neurotransmitters diffuse across the synaptic cleft, and as a result we will see that the majority of these neurotransmitters will end up on that part of the postsynaptic membrane that sits the closest to the axonal terminal. Here and there we might see some neurotransmitters wander off a little bit further, but we will not see as many neurotransmitters bind far away from that axonal terminal. The more neurotransmitters bind, the longer our chemically gated blue ion channels will remain open, and therefore the more and more graded potentials will begin to generate. Now, as a graded potential forms, what happens is that this graded potential will immediately start traveling away from the source of origin. And it does that along the cell membrane of the cell body and the dendrite. So what you now need to assume is that because so many neurotransmitters bind and we keep generating graded potential after graded potential after graded potential, we're going to see that these graded potentials manage to travel further and further as they get stronger and stronger, because they literally go through a process of what that we call summation. Summation means that one graded potential adds up with another graded potential, adds up with yet another graded potential, and so on. The idea is that due to the process of summation, and I tried to underline that word, the idea is that at the axon hillock, we're going to reach a voltage change of about 15 millivolts, and that would open the voltage-gated ion channels. We will use the next video clip to continue our discussion.